All right. Thank you, everybody, so much for being here this afternoon in the last session of this first day. Sanjeev and I will be your moderators for this biothreat preparedness session, and I'm excited to introduce for the first talk a couple different presenters who will be tag teaming this presentation. Mr. Jamie Ballarashti is the founder and president of ARE Labs, a vertically integrated full-service testing, research and development, and engineering firm specializing in all aspects of aerosol science. And their expertise covers a wide range of pharmaceutical, biological, and chemical aerosol programs. And Mr. Mitch Simpler is a retired managing partner of JB&B Consulting Engineers specializing in hospitals, laboratories, and life sciences facilities globally. Looking forward to your talk. Take it away.
Well, thank you. So today we're here to present to you the use of triethylene glycol as a potential aerosolized viricide, which could be used to inactivate airborne pathogens in occupied spaces. Presenting to you all today, as just announced, will be me, Mitch Simpler, and Jamie Ballarashti. I will present the engineering opportunities of aerosolized triethylene glycol, and Jamie will focus on the science. Note that Jamie and I are both here representing Grignard Pure Company, which produces a product called Grignard Pure. And Grignard Pure is a product that uses triethylene glycol as its active ingredient. So you hear us referring back and forth, but that's what we're talking about. When we're all done in the next 20 minutes or so, it is certainly our hope to answer the simple questions as to how, what, when, where, and why triethylene glycol could be what we propose it to be. So the problem we are addressing is containing and mitigating the spread of airborne infectious diseases and pathogens. Grignard Pure with triethylene glycol could be the solution to the problem. To put it simply, triethylene glycol could solve both the containment and mitigation of the spread of airborne infectious diseases and pathogens. The key issue here is that triethylene glycol could be used on an incredibly wide spectrum of airborne pathogens. It could be applied quickly and easily into almost any occupiable space with the amazing potential reduction of airborne pathogen concentrations of 2 to 3 orders of magnitude. I must note that Grignard Pure with triethylene glycol is currently only approved by the EPA for use as mold odor control. Grignard Pure is in the process of getting EPA approval for the use up to and including continuous application in occupied spaces. This product could be deployed both episodically or on a continuous basis, and is readily scalable and, most importantly, completely safe for people, plants, and animals. Most importantly, triethylene glycol could be used to mitigate airborne pathogens in the physical airspace surrounding a bioaerosol shedder, which no other technology can do, especially in the incredibly short time that triethylene glycol does it. It is now my pleasure to introduce our second speaker, Jamie Ballarashti, otherwise known as the scientist.
Thank you, Mitch. All right. Good afternoon, everyone. My name is Jamie Ballarashti, and I will be presenting efficacy results of the Grignard Pure agent against various aerosolized challenge species. Due to time constraints, I will be going through the data very quickly, but there's a short question and answer period afterwards, and also will be available after the talk and during a poster presentation tomorrow to go through in detail. We know there are a lot of natural processes that reduce microbes in the air over time, and it's important in any efficacy testing to account for these natural losses. Our study design accounts for these losses. And you all are familiar with the various technologies that are currently available on the market. All these methods require movement of bioaerosols, usually to the mitigation device or the kill zone when talking about UVC technologies. So what is needed is a persistent kill mechanism that does not rely on movement of large volumes of air. If you've been to a rock concert or any type of theatrical performance, you have probably been exposed to glycol aerosols and glycol vapors in the past. What if the solution was a glycol vapor that also shows efficacy against various microorganisms? We believe the solution is triethylene glycol. Triethylene glycol is not a new discovery. It has been shown to inactivate various bioaerosols for over 80 years. Here are just a few published articles from the 40s showing the efficacy of TEG against various pathogens, including influenza and bacteria. As Mitch mentioned, this is a collaborative effort to bring TEG and specifically Grignard Pure as a solution for pandemic response and disease spread. All the data I will present were trials conducted in various bioaerosol chambers, but all of them were room-sized bioaerosol chambers. Sampling was conducted over time for both control and GP agent trials to show net log efficacy of GP over time for 9 broad spectrum microorganism classes. The testing was conducted to replicate 3 different methods or ways that we believe Grignard Pure could be used to reduce the spread of pathogens. The first method I'll be talking about is the introduction of a one-time burst of GP into an existing bioaerosol plume. Sequential impinger samples are taken to show the reduction of bioaerosol viability over time, and this data is then compared with the control data to yield the GP net efficacy against the aerosolized microorganism. Scenario 1, as I mentioned, this is aerosolized plume in the chamber, followed by a 4-second burst of Grignard Pure into the chamber. This is testing results against non-enveloped RNA phage virus MS2. The graph shows the controls at the bottom of the graph shows natural decay and die-off, while the top curves show the net log reduction of the trial. As you can see, that the Grignard Pure at a concentration of approximately 0.2 milligrams per cubic meter, which is not a visible aerosol at all. So I would like to stress that. So even though the previous slide showed the fog, these are not visible aerosols. But we can see that by the 15-minute mark, we're showing greater than net 3 log reduction in viability by that time point. And this data was also confirmed by 2 independent testing laboratories. Scenario 2 is similar to scenario 1, but instead of a single burst of Grignard Pure into the chamber, this is a continuous steady state concentration of Grignard Pure is maintained during the entirety of the trial. The concentrations range from 0.05 milligrams per cubic meter to 2.0 milligrams per cubic meter. This is more data on RNA phage MS2. The graph on your right shows the controls without the agent. The graph in the middle shows gross log reduction, and the graph on the right shows the net inactivation due to the Grignard Pure treatment. There are also various concentrations, as I mentioned, and one thing to take away from the graph is that for the various concentrations that were tested at this time, we see that the results are very similar. So we believe that perhaps we could even go to a lower concentration of Grignard Pure and still show a wide degree of efficacy. The last scenario involves aerosolizing the organism into an established low concentration of TEG. So a persistent dissemination of TEG into a room as a mitigation, and then a viral generator, viral generators aerosolizing into the existing TEG. This is also for MS2 data. And what you can see here is similar results previously. We see that the initial, we have anywhere from 1.7 to 2.6 net log reduction from the initial concentration compared to the control, and a maximum of 4 net log reduction over the 120 minutes. But it is very noteworthy to look at the 30-minute time point that all tests shows 3 net log results within 30 minutes, and fairly close to 3 net log results at the previous time point as well. So comparison with EPA testing conducted with ORD in 2021 shows independent testing laboratories have similar results to the EPA ORD laboratory. So this slide documents the averages and ranges for results from the various test facility to show that they are in agreement. And this data here is all for MS2. So all this data I presented is available in a recently published peer-reviewed paper that appears in Environmental Science and Technology in 2023. The paper was published by the scientific team supporting Grignard Pure as a novel method for disease mitigation. All right, so up until this point, I presented efficacy results just for MS2. The next several slides, I will present additional species that have been tested using the same scenario 3. So as a reminder, scenario 3 is the existing TEG vapor in the room, and then the subsequent introduction of the bioaerosol. So the first species I would like to show results for is a lipid enveloped RNA phage Phe6. So this phage is used as a surrogate for smallpox, and is also an enveloped virus, much like SARS as well. So for this graph and all the future graphs that I'm going to show you for the remaining species, you'll be able to see 3 lines on the graph. The bottom dotted line is the chamber controls. The top dashed line is the gross log reduction, and the center solid line is the net log reduction. In this trial, since we are aerosolizing into the space with GP, once you begin aerosolizing into the space, you immediately have kills starting to happen. So what we are looking at to compare that very first time point is we have matched trials where we have the same batch of nebulization stock at a split, and then controls and the GP trial were run at the same time. So that initial 1.6 log reduction, that is 1.6 logs below the maximum concentration, viable concentration achieved in that chamber. So having a concentration of Grignard Pure of approximately 2 milligrams per cubic meter in the chamber resulted in a 1.6 log net log reduction at immediately at T0. Also to note, at 15 minutes, we hit a maximum log reduction at 2.26. I will say that in this trial, this series of trials that we did, we had a fairly low viral concentration titer, so we were hitting our limits of detection at 15 minutes. Next, I will be talking about effects against gram-positive species and also mold spores. This shows efficacy against Listeria, a serious cause of foodborne illness. And results are similar to what we have seen with Phe6. We get an initial 0.8 reduction from the T0 compared to the control. And at 15 minutes, we're already at 2.5 net log reduction by 30 minutes above 3 log. This is efficacy against Staphylococcus epidermidis, specifically methicillin-resistant Staphylococcus epidermidis, which serves as a surrogate for MRSA, which is a prominent hospital-borne pathogen, as you know. Data shows, once again, extremely high efficacy resulting in immediate reduction of 1.3 logs below the control on the maximum viral concentration achieved in the room, and over 4 log reduction at the first impinter sample, which is at 15 minutes. Next, the test was against Aspergillus spores. So spores, as you know, are extremely hardy, and black mold is responsible for respiratory illness and sick building syndrome. So the spores that we used are purified Aspergillus brasiliensis spores. They are fed in with a dry powder disseminator. A known quantity, X amount of the same amount of milligrams were disseminated into the chamber. And as you see, initially we only get a reduction of 0.17 net log compared to the controls. However, by the time we get to the 30-minute time point, we still are showing a respectable 1.42 net log reduction of these extremely hardy species. So in addition to those species that I've just shown you, additional testing has been carried out in an identical fashion for Mycobacterium, Pseudomonas, Klebsiella, and Salmonella. And Grignard Pure shows similar results as it does to the other vegetative bacteria shown earlier. It should be noted that GP has been tested in at least one species from the accepted microorganism hierarchy table shown to the right, and the results on the left show the maximum % reduction achieved in the trial. And much like the other data that was shown, it should be noted that most of these maximums occur within either the 15-minute or 30-minute air sample time point. So in conclusion, our research shows that Grignard Pure has a high level of efficacy against a broad class of microorganisms. And I'll turn it back over to Mitch to finish up with the engineering portion.
Thank you, Jamie. The all-volunteer engineering team, which I was privileged to chair, has published several white papers on the behavior, distribution, and control of triethylene glycol in real-world environments, and tested the use of triethylene glycol in real-world applications to validate the propositions that are described in the white papers. The walkaway from that is that triethylene glycol is a controllable, measurable substance that we can control in real time using commercially available technology. So that the technology we use to disperse it is commercially available, and the sensor technology, which measures the concentrations in real time, is also commercially available. The other key takeaways from the white papers include extensive engineering assessments are necessary for any facility. Existing physical equipment limitations prohibit any real meaningful infrastructure upgrades. Increasing the amount of outside air or fresh air has potential and significant operating cost, capacity, and carbon footprint impacts. Increasing filter efficiency in existing HVAC equipment has significant limitations and can actually reduce total airflow into the occupied space. And filters in and of themselves do not inactivate viruses or kill bacteria. To achieve the same active virus reduction achieved with triethylene glycol within the occupied space would require 90 to 180 minutes at the industry standard of 6 air changes per hour. So what does all this mean to everyone here at Decon? This technology allows for almost any viral or bacterial action. It could be a readily scalable response for both small, isolated breakout events, as well as up to and including large-scale pandemic and national events. It could be deployed on a continuous prophylactic basis to maintain utmost national readiness for the military, mass transit, food supply, critical infrastructure, critical manufacturing, healthcare, institutions, and government, as well as law enforcement. Triethylene glycol could also significantly augment PPE. It can significantly reduce of hospital-acquired infections in places like field hospitals, veteran housing, VA facilities, nursing homes, and the like. The associated mold odor elimination properties of TEG also reduces mold spread and viable mold spores in the ambient air. The how. Triethylene glycol could be easy to use and deploy through both mobile and portable plug as well as plug and play systems. TEG, triethylene glycol, could easily be retrofitted as bolt-on technology into existing HVAC systems. Companies such as ServPro or similar response forces who are already working with the Department of Homeland Security and the National Guard could be used as national stocking locations as well as rapid deployment partners. And interestingly enough, there are 20,000 lighting special effect engineers who have been using this product for over 30 years. They can also be used to deploy the technology in a rapid response. Grignard Pure with triethylene glycol could solve the 5 pillars of fighting emergency infections established by the Lancet Commission. It could be used to stop an outbreak before it starts. It could be used to eliminate the transmission of the disease. It could be used to save the lives of people already with the disease. It could be used to ensure that economic and social burdens are shared. And it could be used to be an integral part of the global innovation and diffusion strategy as a new therapeutic. These are links, and I think you're going to get a copy or have access to the PowerPoint presentation. These are live links that can give you access to the engineering and science papers that have been published for TEG and for Grignard Pure. That concludes our formal presentation. Are there any questions? Do we have any time? Yes. We have a minute.
All right, I'll just project.
If you could please state your name and your affiliation, I would think we have time for 1 or 2 quick questions.
Retired Air National Guard. Thanks for this presentation. It looks extremely promising, but what's the status of this regulatory getting this approved by the EPA? What's the status on that?
So far it's been approved for use for mold odor control. And to answer the question that I think you're going to is when can we expect this to be available on a more broad base? I'm going to defer to my colleague, Mr. Bill Jordan, who is a consultant to GP for the last 3 years, but worked for the EPA for 40. Yeah.
Thanks. EPA has previously approved this product for use during the pandemic on an emergency basis to deal with SARS-CoV-2. And when the pandemic ended, so too did that approval. But the company is pursuing, working with EPA, the approval for public health uses. And as the data that Jamie presented shows, it'll work against a wide range of pathogens. EPA understandably wants to make sure that private companies' research is scientifically solid, so they have been doing their own work. Dr. Ratliff at ORD's tested it, as Jamie reported, and that testing has also been done in another private laboratory. So the upshot of it is, I think that EPA is close to issuing guidance that will tell companies how to run these studies so that they can feel confident about the products really working the way that they're claimed to work. And as soon as EPA does that, this company will be ready to submit its data. We think that data will stand up against scientific standards, and we hope EPA will move ahead expeditiously to approve expanded claims.
Thank you, Bill.
One more quick question.
I think this works now, maybe. Yeah. So
It's
Jenny
working.
Thomas, CDC. I see you have a link here. That was going to be my question. Can you describe any human safety data with respect to the concentrations required to achieve the reductions that you displayed here?
Unfortunately, I don't have my hearing aids in, so I didn't
That
understand.
wasn't loud enough. Quiet.
Let's say the levels you're using to test.
Yeah.
I'm back.
So there are a variety of tests that were run in the 40s, and EPA did an assessment of the scientific database on the toxicity of TEG. They concluded that at 1,000 milligrams per cubic meter, there were no adverse systemic effects. And as Jamie described, the levels at which TEG displays efficacy are in the range of 1 milligram per cubic meter. So there's a very ample margin of safety, margin of exposure between the effective level of TEG and the levels that really don't seem to have any systemic impact.